Without further ado, I'd like to introduce Professor Julian Knight from Oxford. And Julian is Professor of Genomic Medicine at Oxford. He's also Director of the Graduate School of the Medical Science Division of the University. He's also Director of the Doctor's Training Program in Genomic Medicine and many other things as well. Julian, you're very welcome.
Thanks very much, Seamus. It's a real pleasure to be here. I'm very grateful to Claire and the organising committee for the opportunity to share with you some of our adventures in terms of understanding the sepsis syndrome. But I think for those of you that don't work on sepsis, hopefully some of the approaches we're using in terms of big data, machine learning, and the broader themes of this session will be of interest. So in terms of infectious disease, with the recent pandemic, we will be all very well aware of this continuing, ongoing major public health challenge. Fortunately, for the majority of us, when we encounter a pathogen, our immune response is appropriate, and the associated metabolic and other components of that response mean that we contain the infection and eliminate it, restoring some form of immune homeostasis. For some patients, particularly younger patients with underlying primary immunodeficiencies, for example, there may be a complete failure of that response. And in others, it is inappropriate or maladaptive. And somewhat frustratingly, the great man was ahead of us by at least 100 years in terms of this very important recognition that except on few occasions, the patient appears to die from the body's response to infection rather than from it. It's taken us a long time, and indeed it's only in the third consensus definition of sepsis that dysregulated host response has become part of the definition. So if we think about that definition, life-threatening organ dysfunction caused by a dysregulated host response to infection, unfortunately it remains very broad. We have a very sick patient with failure of one or more organs, and we have evidence of infection, but we do not have a biomarker or a measure of that dysregulated response, host response. And therefore, it is something of a catch-all diagnosis. Sepsis syndrome, as defined, is a huge problem. So over 49 million cases in a work published before the pandemic. And in the UK, you'll be aware that it's the most common reason for admission to a medical intensive care unit, and the mortality rate remains stubbornly high. And if you survive sepsis, your outcomes are worse than many cancers, so 15% of patients will be dead within 1 year. Unfortunately, for a long time, pharma have started to move away from sepsis as an intractable problem. There's been a consistent failure of clinical trials of immunomodulators despite good preclinical evidence, for example, from animal models. And I think there is a view within the field that this is largely because of a highly heterogeneous clinical syndrome as the entry point for these trials. So there were no new immunomodulatory therapies arising from a huge investment. I think that COVID has shown us what is possible with a somewhat more homogeneous group of patients. And there is clear benefit in the very sick from immunomodulation, for example, with dexamethasone, inhibition of IL-6 receptors, JAK inhibitors. So essentially what I'm going to be talking to you about today is it possible to move from a highly heterogeneous clinical syndrome to understand disease mechanism and deliver more effective therapies? And underpinning, I think, this morning's session is going to be a recognition that we are now in a sweet shop, if you like, if we're interested in using these new technologies in terms of a bewildering array of ways to get very deep phenotyping on our patients. And this ranges from the molecular to an enormous range of modalities that go up to, for example, clinical imaging and effective use of the electronic healthcare record. So it's multimodal data and recognizing what are the translational opportunities of these huge amounts of information. If we think about the immune response, that is very variable within us all, including both innate and adaptive immunity, and our particular response will be influenced by a whole variety of things, including genetic factors, but also our exposures over the life course, the effects of aging, the effects of comorbidities. And I think increasingly there's a recognition that epigenetics plays a very important part in this memory, both in terms of innate and adaptive immunity, and mediating the interaction with the environment. So multi-omics continues to grow in terms of what we can do. We can look at single cell resolution, we can look spatially within tissues, and although it remains expensive, it's increasingly the opportunity to deploy this at scale. And so if we can start to look at large numbers of individuals, we can start to understand drivers of this individual variability. But I think we're going to hear across the talks how there's a real issue, for example, in terms of the data itself, with data missingness, very high dimensionality in the data, a challenge of how you actually maximize the value by integrating these different data types, and can you actually generate this data in a disease context and do some form of validation to learn from it. So I'm going to describe to you how we've used transcriptomics as a way of subphenotyping the sepsis syndrome. I'll tell you a bit about how genetics may interplay with that, and ways that we're thinking about using these different layers of omic and other information in a more integrated way, and finally how we might move this operationally into a more personalised medicine approach for patients. So I'll go back in time a little bit just to say that an initial study in the UK Genomic Advances in Sepsis study that was set up by my colleague Charles Hines at Barts. And with Charles, we worked to build up a large cohort of patients admitted to intensive care and collected the total leukocyte population and did a number of things, including using transcriptomics. And if we then looked at the most variable genes and did some unsupervised clustering and said, how do these sepsis patients start to group, there did indeed appear to be some structure to the data. So we were seeing, when we did a principal components analysis, so this is looking at major sources of variability in the data. Just under 20% of this variability is represented by the first principal component, and that is broadly splitting these two groups of patients. Each dot represents the data from one patient. And these are all patients admitted with community-acquired pneumonia as the driver of their sepsis. And we were struck by a difference in outcome for these patients based on the two groups, with a poor outcome group that I'll refer to as sepsis response signature 1. And this was more reflective of an immune paresis type of phenotype. And this was work that was done with Emma Davenport and Katie Burnham. Katie then went on to look in patients with fecal peritonitis as the cause of the sepsis. And if we return to that principal components analysis, this is looking at the data and thinking about whether you had pneumonia or peritonitis as a cause with the filled and unfilled circles. And you would think that these are dramatically different etiologies. And yet the variability in the data overall, you can see, is really being driven in terms of red and blue between these two groups. And we've gone on to look in a variety of other settings, and broadly it seems to be independent of the pathogen. We then did some work with Tony Gordon and David Ancliffe in London, who had run a number of clinical trials in sepsis. And this is a small post hoc analysis, but it just gives you an idea of the opportunity. And Tony and David are now leading prospective trials together with others. But essentially, when we looked retrospectively, in the groups that received the placebo, we saw no difference in survival. And yet, when we looked at and assigned the groups based on the gene expression patterns, we found that actually the steroids were probably causing harm in the patients that were having a more appropriate response. So I think this is just a taster of what opportunities there might be for an approach to giving therapy where you base it on, in this case, membership of a particular subphenotype of sepsis. Kiki Cano-Gomez then extended this to think about, rather than discrete groups, can we come up with a likelihood score of being in one of these particular subphenotypes? So this is a score between 0 and 1. And we applied it to a variety of settings, but essentially this is showing you, if you look in COVID, a higher score being more likely to be in this poor outcome, immune-suppressed phenotype. And you can see there's a clear relationship with your WHO criteria in terms of critical, severe, and mild hospitalized cases, and a strong relationship with severity. I think this is probably most informative actually in terms of patients that persist in that subphenotype if you measure it on more than one occasion, and those patients do particularly badly. So what does this mean? So just to give you some insights into that from a more immunological perspective, this is work done by a PhD student in the team, Andrew Kwok. And Andrew did a somewhat expensive experiment because we realized from the transcriptomics that neutrophils were almost certainly important. So we couldn't work with peripheral blood mononuclear cells that had been cryopreserved. Andrew needed to use fresh whole blood and do single cell omics on those samples individually. And he did that for sepsis patients, some healthy controls, and then as a measure of a more sterile inflammatory response, he took patients before and after cardiac bypass surgery. And he did whole transcriptomics on this and also measured cell surface protein expression. And that meant that we could get a really good map of these different peripheral blood cell populations within the blood, and a bit like that principal components analysis, this is just spreading the data. You can see the neutrophils here, different stages of maturity, and then we've got the monocytes here and T cells and B cells. And then we said, how can we start to compare between these two subphenotypes? So we assigned the subphenotype based on a predefined set of 7 genes. And so we could then say what was different in the SRS1 patients versus the non-SRS1 patients. And it really seemed